We will be recording this webinar, so you will be able to access it again later if you'd like. We will send it out to all of you. But please stick around for the Q&A, which will happen at the end of the approximately half-hour webinar. And please send any questions in the webinar, and we will address as many as we can afterwards. It is my pleasure to introduce Chris Mosby, PhD and manager of next generation sequencing for GeneWiz. He will be presenting for you today the evolution of amplicon sequencing, methods, and applications. And without further ado, here's Chris.
Okay, Sam, thank you for the introduction. Today I'll be discussing what's seemingly very popular solution amongst researchers, amplicon sequencing. As you'll see throughout the talk, there are many different applications of amplicon sequencing, and within each application, there are many different considerations that you need to take care of when you're planning your experiments. Lastly, there are quite a few advancements in amplicon sequencing that many researchers are taking advantage of. First, before we talk about this, it's vital to understand the backstory. Sanger sequencing was developed by Frederick Sanger in 1977. This method spurred many scientists to start incorporating sequencing into their research. Shortly thereafter, polymerase chain reaction, or PCR, was invented. Coupled together, scientists could now sequence small pieces of DNA quite easily. The Human Genome Project, probably one of the most significant achievements in human history, has accelerated the need for higher throughput sequencing. In comes next-generation sequencing, or NGS. In the beginning, this method had quite a few drawbacks, including read length. As a result of the high cost of NGS and low throughput of Sanger, the Human Genome Project took over 10 years and $3 billion to complete. The next decade focused on unlocking the true power of NGS. Developers focused on scalability of the NGS. As you can see on the left, there was a steady inverse relationship between the cost and the throughput. The cost was steadily declining while the throughput was going through the roof. In fact, this technology was outpacing Moore's law, shown on the right. This opened up researchers to the ideas of using NGS to answer their biological questions. This is indicated in the sheer number of genomes that have been sequenced, also shown on the right. The Human Genome Project took over 10 years and $3 billion to complete, as previously stated. However, today we could sequence a human genome for about $1,000 in under 3 days, which is very impressive. So to summarize, we saw great advancements through the years regarding DNA sequencing. So where do we stand now? Well, developers were not only focused on continuing and improving the scalability of next-gen sequencing, but also focusing on improving the read lengths. Original systems sequenced less than 50 base pairs. However, today we can achieve over 100 KB. This slide shows the relationship of these platforms with regards to one another and regards to the read length and the output. With that said, you can see some trends emerging here on the screen. First, you'll see where Sanger fits compared to other NGS technologies. Sanger is most noted for its very cheap reaction prices and 1 KB. However, it has very low throughput. Next, you'll see where the most popular Illumina-based NGS platforms are. These platforms are known for very inexpensive cost per base pair sequenced, extreme throughput, but relatively shorter read lengths compared to Sanger and some other NGS technologies. Their read lengths span up to 300 base pairs. These are by far the most commonly used NGS platforms. Lastly, you'll see where the trend is going. With that said, long read platforms like the PacBio SQL machine offer a medium throughput, but extremely long reads. The one downside of these machines is the sheer cost compared to the Illumina-based technologies. So the question remains, why do we care about all this with regards to amplicon sequencing? Well, all these factors need to be considered in order to determine how to generate and sequence your amplicons of interest. This slide shows some high-level applications of amplicon sequencing that can help you plan your next experiment. I'll discuss each of these one by one in the coming slides. However, this is only a small snapshot of the most popular applications that I've seen in the field. There are many, many more applications for amplicon sequencing not discussed in this webinar. Take some ideas you would apply amplicon sequencing to help answer your biological questions. So the first application I'll talk about is variant detection. So in layman's terms, what do I mean by this? Well, researchers want to determine if there are differences between the known reference sequence and what's specifically in their samples. One way to do this is to PCR the region of interest and sequence it. Considerations need to be given to the type of variant being analyzed, or what you would like captured in the data, what the expected minor allele frequency is, and how many genes or target loci you want to assay. So to jump to the first question, what type of variant are you interested in? Variants come in all shapes and sizes, so to speak. They range from large structural variants, like translocations and rearrangements, to small SNPs, or single nucleotide polymorphisms. Larger, more complex variants are best suited for shotgun sequencing or whole exome sequencing. Smaller variants, like SNPs, are easily captured in Amplicon sequencing. So if you're interested in structural variant discovery, it's wise to possibly avoid Amplicon sequencing. Amplicon sequencing can, however, detect some known structural variants. Small SNPs and indels are typically captured with shorter reads from both Sanger sequencing and Illumina-based NGS. However, the ultra-long reads of the PacBio sequel, for instance, can be used to detect very tricky indels or even gene rearrangements that can span longer than the aforementioned Sanger and Illumina reads. Moreover, longer reads are ideal for what's called phasing. In layman's terms, this is an assay to determine if a certain variant is located on the same allele for multi-chromosomal organisms. And this is only achieved if the read length can span those types of variants. The next question researchers need to ask is, what is the expected minor allele frequency? So this slide shows two types of variants, germline and somatic variants. Germline mutations are present in all cells in an organism. They are passed on generation to generation. In a diploid organism with two chromosomes, variants are either homozygous, as in 100% of the reads, or heterozygous, 50% of the reads. This opens the door to use Sanger sequencing based on its sensitivity. Sanger can detect typical variants that have a minor allele frequency as low as 30%. So when does this typically occur? Well, shown on the right are what are called somatic variants. These variants are not found in every cell of the body. The most common type of somatic variation is oncogenic, or cancer-related. The minor allele frequency for somatic mutations can be well below 1%. To obtain this level of sensitivity, researchers typically rely on NGS for very deep sequencing. The high throughput nature of NGS can also be used for screening germline mutations in high sample numbers, something that may not be logistically feasible with Sanger sequencing. So that alludes to the next question, is how many genes or targets do you want to screen? If your plan is to screen only a handful, perhaps it's easy to PCR each individual locus and perform Sanger sequencing. However, if there are quite a few targets, a multiplexed PCR may be the best approach. This combines many primer pairs in the same PCR reaction. The resulting mixture can then undergo NGS. So give consideration to manual versus software-guided designs for this multiplex PCR. You could certainly save money designing your own panel, but there are inherent risks with this. This is why there are so many vendors that offer their own software and library prep kits that you could buy for multiplex PCR. If you do want to do your own design, please do give consideration to validating your design. Many researchers will perform individual PCR reactions with the primer sequences they're using in their multiplex PCR pools to prove that the loci are being amplified. Moreover, they're also performing the multiplex PCR, and you need to take into consideration regions that may have dropout due to some various biases when you combine all these primers together in a multiplex PCR. Of course, with any study, I'd recommend validating the results after you do your NGS with many other different types of assays like Sanger sequencing or qPCR. Okay, so we powered through variant detection. The next application that I'll be talking about today is looking at clone verification. So for this webinar, when I talk about clone verification, I'm referring to determining the outcome of genome editing assays in particular. By now, most, if not all of you should have heard about CRISPR-Cas9 mutagenesis. The power of this approach is unparalleled. The accessibility is becoming commonplace. After doing your mutagenesis reaction, researchers typically have to confirm the samples have the correct mutation. There are many ways to do this, which is shown on the screen, but most commonly through PCR amplification and target locus sequencing, which could be done many different ways. Primarily, I'll compare Sanger sequencing with next-gen sequencing. Okay, first, to screen genome editing clones, many researchers choose Sanger sequencing. So why do they do this? This is because they presume their clones are homogeneous. What that means is all cells have the same mutation or lack thereof. This is a very cheap and fast way to screen clones. However, how would a researcher really know if their clone is truly homogeneous with Sanger sequencing? Well, some researchers particularly don't care if it doesn't matter for their studies. However, some do, and if so, what do they do? So this provides an illustration of where Sanger sequencing may fall short if researchers want to find minor allele frequencies that are very, very small. Consider a sample that was thought to be truly homogeneous. If the majority of the cells had the mutation, the Sanger trace will look like the one on the right. However, you see this clone has a small percentage of cells that do not have the desired mutation. So the question you have to ask the question, is this background in your trace, is it real or is it a false positive? So with Sanger sequencing, you typically can't tell. Another issue is if you try to sequence highly heterogeneous clones with Sanger sequencing. Shown on the left, if there are a bunch of different variants throughout the reference sequence, the Sanger trace is going to be all jumbled and basically uninterpretable. The case on the right, if there's a relatively low minor allele frequency, you won't be able to know if this is background or be able to quantitate this variant. So one way around these types of situations is to use a method shown here on the screen. So this would involve looking at heterogeneous clones with Sanger sequencing in a method called TA cloning. This method relies on polymerase during a PCR reaction, amplifying the target locus, adding A tails to the product. This product can then be ligated into a vector with T overhangs. This mixture is transformed into bacteria, plated, and each growing colony has one sequence. The DNA needs to be extracted from said colonies, and then sequencing can happen via Sanger. As you can imagine, this may be quite labor intensive, and there's only a finite number of colonies that you'd want to pick, prep, and screen. So one alternative to this TA cloning and Sanger sequencing of heterogeneous clones would be using NGS. Instead of ligating the PCR product that you would get after you amplify to a vector, you can jump directly to the NGS sequencing. The other advantage of NGS is that many samples can be sequenced together in the same reaction. This makes the process scalable and cost-effective. Moreover, the analysis of NGS data is becoming easier and more commonplace to do in your lab. This question may arise, how can you sequence many different samples in the same run with NGS? Molecular barcodes allow the sequencing reads to be deconvoluted after the sequencing run. One way to add these barcodes is shown here. You PCR amplify your target locus. Next, special DNA adapters are ligated onto the ends of your amplicons. These adapters contain said molecular barcodes. One limitation of this strategy is that you are limited by the number of barcodes present in these off-the-shelf kits. For example, Illumina only has 384 barcodes. This really limits the number of samples that you could sequence in the same sequencing run. One way around this is shown here. Instead of relying on the adapter ligation step to add your barcodes, you can add them directly to your PCR reaction. These barcodes are engineered in your PCR primers and incorporated in the template for sequencing. In theory, you could design as many of these barcodes as the sequencing read length will allow you. In particular, our lab has designed over 10,000 of these barcodes. So in theory, 10,000 samples can be sequenced on one sequencing run, which makes this very scalable and cost-effective. So moving forward, we talked about variant detection and clone verification. The next topic or application I'll talk about is taxon identification. In short, what this means is it's finding out what types of species are in a given sample. So of course, there's many different ways to do this. One example that I'll give is looking at the 16S gene. But there are, of course, many other genes that you could look at for taxon identification. The 16S gene is a highly conserved gene across all bacteria. The advantage of using this gene for identification purposes is that it contains both highly conserved regions for primer design, flanked by highly diverse regions that are specific to each taxa. Sequencing these hypervariable regions allows researchers to determine what species is present based on a reference database. One example is shown below. This is a plate of bacterial colonies. It's assumed that each colony is only one type of species. What researchers typically do in these scenarios is to amplify the whole 16S gene in two overlapping amplicons. These amplicons are then subjected to Sanger sequencing. Sanger here is employed because there's only one predominant sequence in the sample. It's fast and it's very cost effective. So that alludes to my next question. What happens if you have a mixed sample shown here? So I give a few examples below. On the left is a mouse gut, which is presumed to have many different types of bacteria. And on the right are some environmental samples that could have a wide array of bacteria, like a soil sample or a water sample. So how do you identify the bacteria in these samples? Well, one way would be the laborious TA cloning and Sanger sequencing described in the previous slides. However, this is typically not a feasible option based on the overall diversity of the taxa within these samples. Researchers typically focus on the most hypervariable regions of the 16S gene. Here, we're amplifying the V3, V4, and V5 regions in two overlapping amplicons. These are very, very common regions for taxon identification. These amplicons are then subjected to NGS and downstream data analysis. To note, we did focus on V3, V4, V5, but many researchers focus on other hypervariable regions that could be leading to better results based on specific taxa that they're interested in. So there are also some interesting cases where canonically well-defined hypervariable regions would not identify certain species or even subspecies of bacteria in a mixed sample. Below is an example of an underwater heat vent where extremophiles may be present. If said extremophiles do not have a well-defined hypervariable region for 16S analysis, how do you go about analyzing this? So one way would be to amplify the entire 16S gene and sequence this using long-read NGS technology like the PacBio sequel, which can have read lengths, like I mentioned in the previous slides, up to 100 KB. Okay, so the last application that I'll talk about is library screening. This could mean many different things to many people. So with that said, I'll give some very specific examples of what I define as library screening. So the first example is shown here. So our bodies have our own natural antibody libraries that constantly change over time. So researchers are very interested in learning how our immune response changes to certain stimuli, such as drug treatment, different disease states, etc. To do this, next-generation sequencing is typically employed. The advantage of this approach is that it's relatively fast and very, very sensitive compared to other methods of analysis. So how do people use next-gen sequencing to analyze the natural repertoire? So most researchers are interested in knowing how the expression of different antibody genes change over time. Therefore, starting from total RNA, researchers can use gene-specific primers to reverse transcribe antibody genes. Then a PCR can be performed of the resulting first strand of cDNA to produce a product that could be sequenced on next-generation sequencers. Since these genes are relatively small and the complexity of these libraries is very, very large, Illumina sequencing is typically employed. However, there are cases where researchers would want longer reads, such as alternative splicing. PacBio has recently released an application called IsoSeq, which uses an adapted library prep version to sequence long pieces of cDNA. So another library screening method is called phage display. This method is an in vitro approach that allows researchers to determine binding of a library of antibodies to a known antigen. Sequencing of the encoding DNA is how it's done. Prior to long-read technology, researchers were limited to reads that were only about 1,000 base pairs long. Researchers would typically amplify the heavy and light chains individually and then perform either Sanger or Illumina-based NGS. The caveat with this approach is that the link between the heavy and the light chain is broken since they're not sequenced in the same molecule. Therefore, the pairing of the heavy and light chains is determined by frequency, which is an imperfect science, so to speak. To counter this issue, newly developed long-read sequencing can certainly be employed. Instead of individually amplifying the heavy and light chains, the entire locus can be amplified and subsequently sequenced. This does keep the molecular pairing of the chains since they're on the same molecule. However, consideration has to be given to the throughput. So PacBio sequencing has much less throughput compared to Illumina chemistry. Therefore, it cannot assay as highly diverse samples. So if you're doing phage display, you may want to consider looking at more mature rounds of panning compared to if you're using this with Illumina chemistry. Lastly, I'd like to circle back to popular CRISPR technology that I talked about in one of the other applications. So researchers are also using CRISPR to perform functional screens. A library of guide RNAs can be transfected into a cell line. A selection can then be performed, and the researcher can then choose clones that show a particular phenotype. The guide RNAs can then be sequenced from this clone to determine their frequencies. All in all, the sequence of these guide RNAs will refer back to the specific genes, which could infer their function. So how researchers do this is they amplify these guide RNA pools based off of conserved regions outside of the variable regions. So to summarize this webinar, we started the webinar discussing how far DNA sequencing has come over the years. Sanger sequencing has become much more scalable and cost-effective. However, there was a growing need for even higher throughput. This is where NGS came in. Even this relatively new technology has seen a major overhaul in the past few years. There are many applications that we discussed that can employ various amplicon sequencing strategies. I hope this got the audience thinking of many other ways that they can use amplicon sequencing to answer their biological questions in the near future. Lastly, there have been constant advancements in the field of genomics. Researchers can now barcode over 10,000 samples in one run, reads span over 100 kb, and once thought of as complex NGS data analysis is now commonplace in many labs, and there's many open source software researchers can use to analyze NGS data. So with this said, I'd like to thank everyone for attending this webinar. I'm happy to help answer any questions from the audience at this time. So I will ask if you can add your questions into the Q&A section of the chat box, and I will address as many as I possibly can. For those that I do not get to during this time, I could certainly follow up with you offline. Okay, so the first question coming in, do we need to validate NGS results using Sanger sequencing when we are targeting multiple genes using multiplexed assays? It's a great question, and as I described during the talk, it is a good idea to validate your NGS data. With multiplex PCR, there's many different types of validation that you could do. First part of the validation would be prior to actually doing the multiplex PCR, but after you do your NGS, it's certainly a good idea to validate any interesting variants that you find. And researchers, depending on the minor allele frequency, either choose Sanger sequencing or qPCR. So the next question coming in is, what is the maximum amplicon size you can sequence using NGS technology? So that's a great question. So Illumina-based chemistries, they have shorter read lengths, up to 300 base pairs from each end of the amplicon. So this can really analyze amplicons up to about 500 to 550 base pairs. If you transition to a PacBio sequencing, do keep in mind the lower throughput, but the read lengths of PacBio are very long. On average 10 to 15 kb. So our lab has tested amplicon sequencing up to about 10 kb. With that said, the polymerase has a finite read length. So for 10 kb amplicon, the polymerase will only go around a few times. For a shorter amplicon, like say a 2 kb or a 1 kb amplicon, the polymerase can go around a few different times, which would then improve the data quality seen from a PacBio run. So another question would be, how accurate is PacBio sequencing detecting small differences such as SNPs? That's a great question, and the accuracy of PacBio is determined by two different factors: the intramolecular coverage and the intermolecular coverage. So if you're doing amplicon sequencing, researchers typically focus on what's called the intramolecular coverage. So this is the polymerase looping around the same molecule many times. And now if the polymerase can loop around about 10 times, this would give what will be 99.9% accuracy, which is very equivalent to Illumina chemistry. Now, if you're sequencing longer amplicons, like a 10 kb amplicon, the polymerase may not be able to loop around as many times. So you may want to bin your data to focus on the longest read lengths for these types of applications to enhance the data quality. Next question from the group is, you spoke about phage display library screening. Can I use a similar approach for screening aptamers, which are quite small? The answer is certainly. So one advantage of NGS is that sequencing starts right after the sequencing primer binds, and the sequencing throughput is very, very high. And in my opinion, Amplicon, my understanding, aptamer libraries have or can have quite high diversity. Researchers have canonically used Sanger sequencing. However, it may be a little bit tricky with aptamers because Sanger sequencing does start after a few base pairs after the primer binds. And if I know aptamer libraries correctly, the insert size is very small, so Sanger sequencing may not be an option. So can you elaborate a little more on the limitations of PacBio sequencing? Sure. So right now, the really good thing about PacBio sequencing is that the read lengths are very, very long. However, there's some things that you need to consider. The throughput. So right now, each chip on the smart cell of the sequel is about 350 to 500,000 reads. This is in comparison to a MySeq run, which could generate about 15 gigabases worth of data. So it's much higher, magnitudes of data higher than the MySeq. Data quality on the MySeq is very, very good. When you're screening longer amplicons, you have to worry about the data quality on PacBio. So you may want to bin your reads and focus on those slowly. Another caveat with PacBio is the per base pair sequence is a little bit more expensive than Illumina chemistry. So with something like phage display, researchers typically rely on looking at panning, round panning that are towards the ends of their experiments where the diversity is not very high. So one more question would be, I have a de novo genome assembly, and I've used short read technology, but I have a lot of gaps in the assembly. Can I use PacBio long amplicon sequencing to plug those gaps? So really with PacBio, there's two ways that you can improve that assembly. So you can use PacBio as a shotgun genome sequencing approach and do more of a hybrid type of assembly using short read and long read chemistries. Another way is you can use the fixed sequence and do a PCR amplification of that perceived gap. That may be a little bit tricky because you have to align these contigs together, and you don't necessarily know the size of that gap. So it may be a little bit tricky that way. So another question coming from the group is, what typically is the accuracy of Illumina paired end sequencing? This is a great question. So the way Illumina accuracy is calculated, it's based off of the actual read length that you're doing. So for instance, and it's also depends on the platform. So if you look at, say, the HiSeq and the MySeq, which are the two most popular platforms, 150 base pair paired end reads, the quality that you would get is typically 8 of bases that have a greater than Q30 score. And a Q30 in layman's terms is 99.9% accurate. So as the read progresses, the quality towards the ends of the reads drops. as well as read 1 versus read 2. So read 2 does have a little bit lower quality compared to read 1, and the back end of read 2 does have a little bit lower data quality.
All right, everybody, that brings us to 12:30 and wraps up our webinar. I want to thank Chris for presenting such a great webinar for us all, and thank you all for coming. Again, please feel free to keep adding some questions. We'll keep signed on for the next 5 minutes, and we can kind of get back to you guys offline if you have any other additional questions we cannot get to. Otherwise, thank you all for coming. Have a great day, and if you have any further questions, please do not hesitate to contact us or Chris at the credentials listed here or on genewiz.com.